So now we're going to take a look at the structure-function relationship for red blood cells. So let's start with what the purpose of red blood cells is. What do they do? Red blood cells transport oxygen. That is really their primary purpose. A secondary function is to transport carbon dioxide, and we'll get to that in another lecture. For now, we can just think of their primary function as to transport oxygen. In cellular engineering, you design the cell for its function. That's the structure-function relationship. For instance, in a muscle fiber, we have a very elongated cell filled with myofibrils, and that makes sense if you're thinking about contraction and shortening the cell. Here, our goal is to transport oxygen. So as a red blood cell, we would want to be designed to maximize the amount of oxygen we transport. So how do we do that? Well, it turns out that there is a special molecule within each red blood cell that actually does the transportation job, and that molecule is called hemoglobin. So hemoglobin is actually a pretty large protein. I'll show you sort of an image of it in a moment. But it is a large protein, and when I draw out hemoglobin, I kind of draw it out like this. I'll take 4 diamonds and overlap them, as shown. And that is because hemoglobin is actually made up of 4 different subunits. And at the center of every subunit is something called a heme group. And that heme group includes an ion. Not just any ions, but specifically iron. So Fe is the chemical symbol for iron. So hemoglobin is going to be made up of 4 subunits, each with an iron atom at its center. So hemoglobin is actually responsible for doing the transportation. And you can think of hemoglobin like an Uber car or a taxi car or like a Lyft driver. And you can think of these diamonds as seats. So if you have a hemoglobin molecule with 4 seats, then it could transport up to 4 oxygen molecules. In fact, we can almost repurpose our diagram of hemoglobin to look like this. So here I'm drawing a hemoglobin molecule, and there are 4 quadrants. Each quadrant represents one of those subunits. So these are equivalent to each other. And that means each quadrant can hold an oxygen molecule. So if I have, go ahead and draw 4 oxygen molecules, then I could load them up like so. So one hemoglobin molecule can transport 4 oxygen molecules. So if a red blood cell's job is to transport oxygen, then doesn't it make sense that you would want to pack as much hemoglobin in there as possible? It should. Consider this scenario. I have 10 hemoglobin molecules. How many oxygen molecules could I potentially transport with 10 hemoglobins? 40. What if I wanted to transport 4 million oxygen molecules? Then I would need 1 million hemoglobins, because remember, each hemoglobin can transport 4 oxygens. So I'm going to come down here and just show you for a moment what hemoglobin looks like. You are not responsible for knowing all of the nuances of hemoglobin. I want you to know that there are 4 subunits, and that at the center of every subunit is an iron atom. And the reason why I want you to know that iron is important is because iron is required to build hemoglobin. So let me say this. Let's say that I have 20 iron ions available. How many hemoglobin molecules could I make? Well, I could subtract 4 to make 1, and that would leave me with 16. Then I could subtract another 4, and that would leave me with 12, so now I've made 2, etc., and you'll realize it's really just 20 divided by 4. In other words, I could make 5 hemoglobin molecules. So what happens if I'm limited in my iron? I don't have enough iron. Then I can't make all the hemoglobin that I really want to be making, which will limit my ability to transport oxygen to my cells. An analogy to that limiting nutrient is this. Let us say that I ask you to make 10 cakes, and I give you unlimited flour, as much flour as you want, but I only give you 2 eggs. And you go, I can only make 1 cake with 2 eggs. To make 10 cakes, I need 20 eggs. And I said, nope, sorry, only 2 eggs available. That's all we have. Well, you're not going to be making 10 cakes, even if I want 10 cakes. You're going to be making 1 cake. And until you get more eggs, you can't make any more cakes. The reason why that's important is because there is a disease or a disorder known as anemia. Anemia is when your hemoglobin levels are too low, because either your red blood cells are lysing, or maybe you're not even making enough hemoglobin. So there's a couple different, causes of anemia, but one of the most common causes of anemia is going to be not enough iron available to synthesize hemoglobin. So this is sometimes called iron deficiency anemia. So let me go ahead and write some of these terms down because they're important, and I want to make sure that you've got them in your notes. Okay, so the concept of anemia, which by definition just means low hemoglobin levels, is explored a lot more in Lab 15. And I just wanted to put out there that iron is required in order to make hemoglobin. And if you're limited on iron, then you're limited on the amount of hemoglobin that you can make, which means that you're not going to be making as much hemoglobin as you might want to make. But let us assume for a moment that the amount of iron is not limiting, that we could make as much hemoglobin as we want. So let's say that I have a hematopoietic stem cell. Here's my hematopoietic stem cell, and it gets the chemical signal from the hormone erythropoietin, that's EPO. And so as a red blood cell, its job, of course, is to transport oxygen. If I want to transport, a billion oxygen molecules at a time, or up to a billion oxygen molecules, then I would need 250 million hemoglobin proteins. Now, hemoglobin is actually a pretty big protein, so that's not a small ask. That's actually kind of a big deal. So let's go ahead and assume that we can make 250 million hemoglobin molecules. And through this process of differentiation, don't draw this out, by the way. I'm really just making a point here. I don't want this in your notes as something kind of permanent. If I start making a bunch of hemoglobin, then my cell is going to get really big. So I'm going to go ahead and assume, here's my nucleus, I'm going to assume that each of these red dots is going to be a hemoglobin. Now, hemoglobin is red. It's red because iron is red. So it's actually what makes the red blood cell red, which makes your whole blood red. So all of this is due to the fact that iron, when it's oxidized like this, is red. So each one of these dots represents hemoglobin, and I'm going to say that we're going to put so many in here. That the cell gets really big. So the cell is now really big. Okay, that's a problem. It turns out that when a red blood cell gets too big, it's not able to do its job effectively. A lot of people think that is because red blood cells can no longer fit through capillaries. But, I mean, we could just re-engineer our capillaries to be bigger, right? I mean, from an evolutionary standpoint, that would make sense. So it turns out that that's not so much the problem. There's actually a bigger problem. So this is not going to be a workable design. Let's take a look at what a red blood cell actually looks like. And then we'll talk about why that form works and how we get there through differentiation. Red blood cells are not your typical cells. They are flat, so they're not spherical, and they have what is called a biconcave shape. So there's the biconcave shape there. So if I draw a cross section, and you can draw this with me if you'd like, you might draw almost like a little figure 8. And it almost looks like somebody took a sphere and then deflated the center. They're also small. So from a previous lecture, you might remember that red blood cells are pretty small, 5, 6, 7 microns long. That's actually very small for a cell. So they're small, they're flat, and they're biconcave. Also missing from this picture, do you guys notice there is no nucleus? It should be in the center there, right? And it's gone. So there's no nucleus, and there are some other organelles that are missing as well, including mitochondria. So we have a red blood cell that is missing some of its most important stuff, which is kind of fascinating. But it's a sacrifice the body is willing to make in order to optimize a red blood cell for its function. And that is what we mean by the structure-function relationship. This idea that we can actually change the structure or modify the structure so that it's optimized for its function. And here we have a cell willing to give up like the soul of its identity, the nucleus, and be like, all right, why? Because it takes up too much room. So instead, the red blood cell is like, wait a minute, if I get rid of this nucleus, then I have extra room for more hemoglobin. So now that I've kind of shown you the punchline, what the red blood cell looks like, let's come back up and figure out, well, how the heck did we get here in the first place? When we go through differentiation, this is what actually happens, so go ahead and draw this down. The cell is going to synthesize a bunch of hemoglobin. I'm going to put up the chemical symbol for hemoglobin, just so that you recognize it. But it needs to synthesize hemoglobin at the same time that it's actually getting smaller. So the hemoglobin will make it more red, so these cells end up looking more red. But then it needs to kick out bulky organelles. So what are some bulky organelles that it's going to remove or degrade? The two primary ones are the nucleus and the mitochondria. And so what we have is a cell that is becoming more red and smaller. It initially will have, part of a nucleus in there. And then finally it gets rid of its nucleus and it achieves its final shape of a small red blood cell. Alright, so that process of going from a generic cell here and morphing into its final product is called differentiation. So what we've diagrammed here is the true differentiation of a red blood cell. So let me go ahead and show you an image of that. So this is what we want to focus on here. You can see the generic looking cell. You can see it's getting smaller and redder, and eventually here it shows the nucleus being ejected, but in reality it's more like degraded until we get a flat small cell. And look at how small it is. Here's a neutrophil, that's a white blood cell. And look at this, this is actually part of a megakaryocyte over here. Okay, so why does a small biconcave cell maximize its ability to do its job? So that's kind of a second question. I mentioned already that we don't want this, right? We said nope, 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 none to this. This is bad. Too big. We want small. And we want thin. But why? So let's talk about that. Let's start with what mature red blood cells are or lack. Mature red blood cells are going to be small and what's called biconcave, and then they're going to lack many of the bulky organelles, including mitochondria and the nucleus. This second part right here, this strategic elimination of those organelles, is so that the cell can make more room for hemoglobin while still maintaining a small biconcave shape, which is needed for reasons we have not yet discussed. Okay, so again, getting rid of bulky organelles frees up space. We can put more hemoglobin in there and still maintain our small size. So now let's get into the more difficult question. Why does it need to be small and biconcave? What's all that about? So in order to understand that, we have to get to something called surface area to volume ratio. It's a totally different concept that actually comes back in multiple forms in anatomy, not just here. But again, it's a concept called surface area to volume. Okay, surface area to volume ratio. Let us say that I have a sphere. So I've got my sphere here, like so. The volume is going to be all the space that sphere takes up. And if my sphere had a radius of 1 without units, then my volume would be like 4.2, whatever it is. Let's say it's 1 centimeter radius. This would be centimeter cubed. By the way, you don't need to write any of this out. The math and whatnot is not needed. The surface area is basically me taking like wrapping paper and trying to wrap up that sphere. And I don't know if you've ever tried wrapping a ball before, but it's actually quite challenging. And imagine for a moment that you have like shrink wrap, not actual wrapping paper. So the sphere actually has not very much surface area for the volume. It's one of the most favorable in terms of packing a lot of volume in for not too much surface area. So the surface area here is 12.56, whatever, centimeters square. Well, let's make this big. So now instead of 1, this is going to be a radius of 2. Obviously it's much bigger. How much bigger? Well, the volume has jumped up to 33.5 centimeters cubed. That's a pretty big increase, right? Like an 8-fold increase almost. Did the surface area also increase 8-fold? And the answer is no. If we were to increase 8-fold, we would expect basically 12.5 times 8, which would be over 100. But the surface area does not take 8 times as much wrapping paper. It takes about 4 times as much wrapping paper. This tells us is that what is called the surface area to volume ratio, the relative proportion of surface area to volume, becomes more heavily weighted to the surface area the smaller you get. So when you get small volume has more surface area relative to the volume. Not only that, but we can also take that sphere and imagine it's a cell, and we can start changing what we're doing with that sphere. So consider this idea. I'm going to duplicate that here. This is my sphere. And now I'm going to take this sphere and I'm going to start pulling out some of the cytoplasm so that it now looks like this. let's say that I kept the volume the same. I just changed it so that I made the surface area really, or the outer surface very undulated. This would have a much greater surface area. So undulations increase the surface area. This is kind of a key piece here. Okay, to maximize the surface area then, relative to the volume, I want undulations and I want a small volume. I want a small cell size. Again, I still haven't talked about why this matters, but it is definitely something that I want. So to maximize the surface area to volume ratio, I am going to keep a small cell size, so small volume. That means a smaller cell. And I'm going to have undulations. In other words, I'm going to move away from just the nondescript spherical cell that I so typically see. All right. The red blood cell wants to maximize the surface area to volume ratio. In fact, it's almost required. Why? Because it increases the ability of oxygen to actually move from the plasma into the red blood cell. So why would you maximize surface area to volume ratio? What advantage does this give you? It helps you with transport. So now I'm going to use some analogies and examples to explain why maximizing your surface area actually will increase the amount of oxygen that diffuses across the plasma membrane. And I'm going to start with an analogy. We're in a room here, right? So I'm actually in a room in my father's house, and there are 4 walls and there's a ceiling. So I'm going to go ahead and draw kind of this schematic of a classroom or some room. And so there's the back. Here's my bench. Here's where I am, my chair. Mickey Mouse is behind me. Okay, so there we are. Now I am going to release 1,000 fruit flies into this room. But I have special wallpaper. I have wallpaper that doubles as fly paper. And so what I'm going to do is I'm going to line all of these walls with this fly paper, special fly paper. So I'm going to put up wallpaper that is actually fly paper, so it's sticky. And when a fruit fly flies into this wallpaper, it's going to get stuck and not move. So I'm going to line all the walls, and you know what, for good measure, I'm going to also cover the ceiling and the floor. This is all my fly paper here. Okay. How long is it going to take me to capture 1,000 fruit flies? So we've got these little fruit flies. Kind of looks like a Harry Potter snitch there. But obviously not drawn to size, or I'd be running out of here screaming. But we've got these fruit flies, and what they do is they fly, right? They do this, they kind of buzz around, and then the idea is they get stuck, and then they can't move. So how long is it going to take to stick all of those flies? I don't know. I would probably say maybe a couple of hours before I captured every single one, because quite a few of them are going to keep buzzing around like here in the center of the room and not go to the outside of the wall, right? So you can think of this room as like a cell, and the plasma membrane is the wallpaper on the ceilings, on the floor, and the flies are like nutrients or something you want. And how many do you capture? How long does it take to capture all of them? Well, it actually takes kind of a long time, because in many cases you have to wait for a fly to run into a wall. Let's say that I have a lot more wallpaper that I can use. And so I decide that I'm going to remove myself out of this room, and I'm going to start adding wallpaper in different spots. So I'm actually going to do kind of a top-down view. So you can see kind of what I do with my wallpaper here. Here's my seat. Here's my chair. There's my backboard right there. And what I said before is that I would line the walls with wallpaper. Now what I'm going to do is I'm going to take I'm going to take that roll of wallpaper and I'm going to start going back and forth like that. And now if I have a fly, tiny little fly in there, and it gets released into this room, it can't go very far before it gets stuck. How long will it take me to capture 1,000 fruit flies if my wallpaper is, like 6 inches from another wallpaper? 5 minutes max, right? Because the fruit flies have nowhere to go before they run into wallpaper and then they get stuck. So in the same way, if I take a cell and I start doing the wallpaper design on that cell, I'm going to increase the likelihood that that part of the plasma membrane is going to interact with something I want. And what I just drew might look a little familiar to you. This is a cell from the small intestine. And so if you have had anatomy, you will recognize these as microvilli, these little finger-like projection. Their only goal is to increase the surface area so that when you come in contact with glucose or whatever else you want, you are more likely to interact with it and therefore be able to transport it across. So we actually see this theme multiple places in our body. It's not just in red blood cells. But by increasing the surface area relative to the volume, I increase the likelihood that I'm going to run into that oxygen. And therefore, the likelihood that I'm going to get that oxygen to move into the cell where it will stick to a hemoglobin and be transported around the body. So what our bodies do is decide to sacrifice volume, keep the cell small. The biconcave shape ends up increasing the surface area. So it's not quite so undulated like a small intestinal epithelial cell, but the idea is the same. Okay, so we covered a lot of different things here, and I just want to basically make sure that we have a pretty good understanding of how this all fits together. So let's start. Goal. The goal of a red blood cell is to transport oxygen. How is it going to do that? Well, it really needs two things. First of all, it needs to capture the oxygen, it needs to get the oxygen into the cell. And then the other thing that needs to happen is we need to bind that oxygen to hemoglobin. So how do we do this? Well, the first thing we want to do is maximize the number of hemoglobin molecules we can shove in a red blood cell. A mature red blood cell is basically just a glorified packet of hemoglobin. So we're going to shove up to 250 million hemoglobin molecules into one red blood cell. Okay, the problem is that as I do that, my cell gets big, 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 because 250 million of a large protein is definitely going to expand that volume. So the cell says, well, that helps me with binding the oxygen, right? More hemoglobin, more oxygen I can transport, but now I'm losing my ability to get it into the cell because my volume is getting too big relative to my surface area. I don't have enough surface area to come in contact with oxygen. So I need more surface area, less volume. What do I do? And you say, well, organelles are expendable. Kick out the nucleus. The nucleus is really bulky and the cell deflates and becomes a biconcave shape. Then we're going to kick out other bulky organelles like mitochondria. Okay, so what we're left with is basically little else but a small biconcave, kind of flattened red blood cell that is just filled with hemoglobin and cytoskeleton and not much else. But there are in fact consequences to losing those organelles. So let's talk about those consequences. First, if you lose your nucleus, what do you lose? You're actually losing your DNA, right? What does your DNA do? It codes for things in your cell that you might need, like to repair. So without a nucleus, the life expectancy of a red blood cell decreases significantly. And here's another analogy I'm going to use. You buy a new car. So you buy a new car, it costs you, $30,000 or something, and you never ever take it in for repairs. So no oil changes, no belt changes, no new tires, no brake, nothing. How long is your car going to last you? Maybe 50,000 miles before the gears start getting stuck because your oil is crummy, or maybe your brakes wear out or your tires go bald, right? So in other words, repair is important. Maintenance, upkeep is important. But if you take your car in and get it serviced every time it's supposed to, and you repair it when it's broken, then your car, depending on model, should last 200,000 miles, I would guess, right? So I won't name which models I think will last longer, but you get the idea. It's going to last a lot longer with maintenance and repair. The cell kind of has the same thing. You've made a cell, it's got all new parts, it's all brand new and shiny. You put it out into circulation and things start wearing down. The cell gets brittle, actually, kind of like leaving plastic out in the sun. It gets brittle. Okay, well then what? Well, if you don't have DNA that codes for the instructions to actually make the repairs, the cell is eventually going to be like plastic that's left out in the sun. It's going to start breaking apart. And so the life expectancy of a red blood cell is actually about 4 months. Now, what you guys need to know about this is that it is the loss of the nucleus that ends up causing that short life expectancy. Okay, so again, no DNA, no instructions, no ability to recreate brittle proteins or degraded proteins and repair your cell, and the cell eventually will lyse. Now, interestingly enough, having your red blood cells kind of go bad and start lysing randomly in your body is not good. You don't want that to happen. So you have a special spot in your body where you kind of like send your red blood cells through in like an audition almost. It's kind of like, are you still in good shape? Yes? Awesome. Go back out to the circulation. No? Then you're going to lyse and we're going to clean up the debris here before you get out into the body. And that location is in the spleen. So the spleen actually is a major site where red blood cells audition for, health and viability. So the spleen has super small capillaries. So typical capillaries might look something like this. Here's a typical capillary. And spleen capillaries are going to be even smaller, right? So if here's a red blood cell getting through, and now it wants to get through the super small one, it's actually going to have to like shrink down and try to squeeze through it. So it has to reform its shape to try to squeeze through a little bit. And so a lot of them they kind of like elongate here and get through. And then when they get out to the other side, they kind of reinflate. And if their cytoskeleton is brittle, then they just lyse. And then the spleen's like, thank you, you're taken out of circulation. You did not pass the test. So spleen takes it out of circulation. The hemoglobin ends up recirculating in the blood, getting picked up by liver to be further degraded into something called bilirubin. But the point being that old, not in great shape red blood cell was removed from the circulation. Okay, so the spleen will kind of audition with super small capillaries. The last thing I want to talk about is the loss of mitochondria. So in addition to losing the nucleus, a red blood cell also degrades its mitochondria. So what is the consequence of degrading its mitochondria? The consequence is that you can't go through a lot of the stages of cellular or aerobic respiration. So remember, aerobic respiration is really how you get most of your ATP, right? And aerobic respiration can be broken down into some stages. We have glycolysis, we have the citric acid cycle, and we have oxidative phosphorylation. The last two cycles occur in the mitochondria, especially oxidative phosphorylation, which is your big ATP maker. It's your big money maker. So when you think about what happens in the mitochondria, like 90% of your ATP is generated there. And the other 10% or less, is generated via glycolysis, which happens in the cytoplasm. So if we get rid of mitochondria, we can't do aerobic respiration. What? Didn't I just tell you that like 2 ATP per glucose is fatal? It is for most of our cells, but think about what a red blood cell is doing. Nothing. It's just floating around. It really doesn't do very much. It doesn't have a lot of energetic needs. It's not doing a lot of work. It's not doing movement. It's not doing fancy transport. Why? Because remember, oxygen can diffuse through the phospholipid bilayer. That's simple diffusion. So therefore, you don't need to do active transport for oxygen. And binding to hemoglobin and releasing from hemoglobin is also passive. It doesn't require any work. And you're not really doing very many chemical reactions, so you don't have very high needs. Red blood cells are not a high need cell. They're not expensive. So they can get by with just 2 ATP. All right, final thing. It's ironic. This is actually kind of like a triple pen here. It's ironic that a cell filled with iron, filled with iron, that also has the best access to oxygen of any cell out there. I mean, red blood cells have tons of it. Can't even use it. So they're sitting on a gold mine of oxygen, and they don't even have the ability to use it because they get rid of their mitochondria. And remember, it is the inner membrane of that mitochondria that has the electron transport chain, and that's what actually consumes oxygen. But they don't have it, so they don't consume any of the oxygen. So they literally are just transporting these oxygen molecules around the body, and they never actually get to use it for themselves. So close, and then they can't use it. All right, so our very last part of this lecture on blood is going to then turn and take a look at platelets and clotting. So we'll save that for the next lecture.